Hi, today we are going to discuss a very important cell organelle which is known as mitochondria, and it is the organelle which is basically very important to discuss about it because it plays a vital role in production of ATP inside our body, inside each cell. So that organelle is mitochondria. Basically, it is a double membrane bound structure. Outer membrane is smooth while inner membrane is convoluted and developed folding inside it. And inner membrane is basically it is solute impermeable membrane because it is having a high content of cardiolipin. So, inner membrane is having cardiolipins, and as well as it is having ATP synthase complex, also known as FOF1 complex, which is um, basically it is passage for protons for generation of ATP. So the main role inside mitochondria also is of FOF1 particle. FO stands for oligomycin sensitive unit. So it's related to generation of ATP through formation of electrochemical proton gradient. When the hydrogen ion or protons pass through them, so there is rotation which goes on inside the particle on FO particle, and this rotation is leading to generation of ATP when the hydrogen ion pass through it. And outer membrane is basically smooth and it's having porin, which is voltage dependent anion gated channels. It is having porins, and mitochondria are basically having double stranded DNA. It is circular, and DNA is passed horizontally through uh, transfer, a horizontal transfer, and it passed on to host nucleus. So the dominant role is caused by mitochondrial DNA. Mitochondria are also semi-autonomous organelles, and they are having their own genetic material. And organelles also, they are also having own genetic material, and they are also having their own ribosome, that is 70S ribosome. So ribosome play important role in protein synthesis, and so mitochondria are also possessing their own protein due to presence of their own ribosomes. So mitochondria is having double-stranded DNA. They are having their own ribosome, that are 70S ribosome. They are having their own proteins. And most mitochondrial proteins are encoded by nuclear genome. It is not possessed by the nucleus in which the mitochondria is residing. It is not possessed by the cell nucleus in which the mitochondria is there, but mitochondria is having their own proteins encoded by them. So, for the origin of mitochondria, the two theories are given, and in the endosymbiotic theory, there are two types of scenarios which is given. One is Archesion scenario. So, there are currently two scenarios going on. What is Archesion scenario is all about? It is that proto-mitochondrial endosymbiont was hypothetically a mitochondrial eukaryote, and which was termed as Archesion. So it was basically a eukaryote, proto-eukaryote, which was termed as Archesion. And there is another scenario for the origination of mitochondria, and that is symbiotic scenario. In symbiotic scenario, same single endosymbiotic event was involving in uptake of a proteobacteria by archaeal cell, and which led to um, generation of mitochondria and evolution of nucleus and compartmentalization of eukaryotic cell. So it is basically saying about the evolution of the cell, which happened by uptake of a prokaryotic cell by a eukaryotic cell inside itself, because it is proven by or it is favored by a hypothesis that it is having their own DNA, own protein. We can say that mitochondria would have been a prokaryote, which was engulfed by a eukaryote, and it is existing there because of their own DNA and protein. And because favoring this scenario is are certain statements, one of which are because mitochondria also divide in a manner like a bacterial DNA does or bacteria does, like mitochondria divide by binary fission. And second one is mitochondria is surrounded by two membranes. Innermost is having similar composition to that of the bacterial membrane. Third one is mitochondria is having own DNA. So it is favoring that it can have been different uh, organism. It would have been a different organism because it is having its own DNA similar to bacterial DNA. Mitochondrial ribosome, enzymes, and transport synthesis are that similar to bacteria. And mitochondria are especially same size as that of bacteria. And next one is that protein synthesis in mitochondria is inhibited by variety of antibiotics such as chloramphenicol, tetracycline, and erythromycin. Very interesting that those antibiotics which blocks the uh, protein synthesis in bacteria are acting the same upon the mitochondria. So it is very interesting to note, and it is also giving a direction to which we can say that mitochondria have been a prokaryotic organism at certain point of time, which is slowly but gradually engulfed by eukaryotic organism, but mitochondria is sustaining itself. That organism, that prokaryotic organism, was able to survive, was able to sustain itself in that manner, and it's residing as a cell organelle. Quite interesting, and we will come up with more interesting topics and interesting details about it in upcoming videos, so please stay tuned.